Hello, everyone. I am Jacob Gillen, a BGSU student majoring in biology, marine and aquatic, while holding a minor in environmental science. The faculty mentor that oversaw my research was Dr. Chris Ward of the Department of Biology. To begin with the presentation, what are microalgae? To start with a general explanation, microalgae are a unicellular microscopic algae. Most of the microalgae that have been discovered are photosynthetic carbon fixers. This means that these organisms are able to convert carbon dioxide into other sugars and oxygen to survive. As part of this process, algae have been found to release up to about 50% of fixed carbon, as well as other elements such as nitrogen and phosphorus. Under optimal conditions, as much as 80% of algal biomass is stored in the form of lipids. This is extremely important because lipids can be extracted from algae and converted to biofuels. To go more in depth on the topic of importance, let's talk about algal biofuels. As many of you may know, fossil fuels are a major contributor to the rising atmospheric carbon dioxide levels. For a more environmentally friendly standpoint, algal biofuels are a promising technology that are known to be renewable, carbon neutral, and free from petroleum-based contaminants. As you can see in the image on the right, the production of algal biofuels requires mass cultures of microalgae. However, these cultures often crash rapidly due to stressors such as nutrient limitation and parasites. These factors make studying the microbiome of algal biofuel ponds increasingly important because of the heterotrophic microbes that are often responsible for much of the nutrient recycling in these systems, which aid in algal culture growth and maintenance. So what are microbiomes? Algal microbiomes are communities of microbes such as bacteria and fungi that are associated with algae. However, just like any other type of environment, there are positives and there are negative interactions between algae and their microbiome. For instance, much of the organic matter in the aquatic ecosystems is too large to be processed directly by heterotrophic microbes. To circumvent this problem, these microbes produce extracellular enzymes that degrade organic matter and other nutrients into smaller pieces, which can then be taken up by the heterotrophic microbes. While the use of extracellular enzymes in aquatic ecosystems has been well studied, the applications of the extracellular enzyme activity assays within the algal biofuel cultures is rather novel. The purpose of my project was to develop extracellular enzyme assays in order to assess the extracellular enzyme activity of the heterotrophic microbes within the algal biofuel cultures. My goal was to develop and run assays that are capable of measuring the extracellular enzyme activity of various nutrient acquiring enzymes in aquatic systems. For this study, I had one main question, which was, how does the extracellular enzyme activity of the algal microbiome within algal biofuel cultures change over culture development? For the methods of the research, I modified microplate methods to assess the extracellular enzyme activity. I used fluorescently labeled artificial substrates and a fluorometric plate reader to track the rate of substrate hydrolysis. If you look at the image on the bottom of the slide, you will notice and see the reaction for one of the enzymes used in this study. MUG is a large organic molecule that cannot be broken down by other microbes. This molecule reacts with the enzyme beta-glucosidase, which is a carbon-acquiring enzyme. After this reaction occurs, the end products are glucose, which can be taken up by microbes, and the MUF-labeled substrate, which fluoresces or omits color, that can then be measured with a fluorometric plate reader. In this study, I used a bacterialized Scenedesmus dimorphus strain from the University of Texas, UTEX 1237. Bacterialized simply means that we had an algal culture and allowed a natural bacterial microbiome to develop. I used MUF-labeled substrates for the following enzymes, which were alpha-glucosidase, beta-glucosidase, and beta-xylosidase. Those three substrates are all carbon-acquiring enzymes. Phosphatase was used, which is a substrate that is phosphorus-acquiring. If you look at the image of a 96 well plate on the right of the slide, we had 5 sample replicates in the green, which contained our sample and the enzyme substrate. Additionally, I prepared wells for the sample controls, which is shown in the orange. These wells contained the sample and sodium bicarbonate buffer. The substrate controls, which are shown in pink, contained the substrate and buffer. And finally, I prepared wells with standards in blue, which contained MUF standard and sodium bicarbonate buffer. If you look at the pink once again, you will notice that there are rows A through D that are used for this. For A, I used alpha-glucosidase. For B, I used beta-glucosidase. For C, I used beta-xylosidase. And then D, I used phosphatase, just to avoid any type of confusion on what the order was. Once this plate was prepared with the proper concentrations and mixtures, I used a TCAN microplate reader to take fluorescent readings every 10 minutes. I did this until I found the plate reader started to record a decrease in extracellular enzyme activity. When the plate reader was done reading the well's plate, I was able to have the plate reader export the data for the plate reading to Excel. If you look at the graph on the right side of the slide, you will begin to notice, with the exception of the substrate alpha-glucosidase, to see the peak extracellular enzyme activity happen around 20 minutes. After the 20-minute mark, you will begin to notice a decline in extracellular enzyme activity. However, with alpha-glucosidase, you will notice that the peak extracellular enzyme activity had occurred around 10 minutes. For all of the enzymes, however, when looking at this graph, the extracellular enzyme activity seems that the activity drops off dramatically at around 30 minutes. This data gives a good timeline to identify peak extracellular enzyme activity and optimizes this procedure for future extracellular enzyme activity assays. In order to make algal biofuel production much more efficient, understanding the algal microbiome is becoming increasingly important. My development and novel application of extracellular enzyme activity assays in algal cultivation will provide further insight into the important roles the algal microbiome plays in the development and the sustainability in those algal biofuel ponds. We will be creating mesocosms with strains of algae commonly used in algal biofuel ponds. As a part of this study, I will assess the extracellular enzyme activity at various points in time over the course of culture development to track changes in extracellular enzyme activity. To determine how the algal microbiome may be affecting culture growth and persistence. Additionally, I plan to use mesocosms to grow multiple algal biofuel strands to understand which strands and their associated microbiomes would be the best suited to produce optimal biofuel yields. And then lastly, if there are any questions about this project, or if you would like to just know more, you can contact me at my email, which is on the slide. Thank you.